Hello everyone, welcome back. So we have this week's assignments, quiz 6, which is going to be due on 4/27 at 11:59 p.m. And that's going to be based off of this coliform bacteria analysis video right here.
Beach closed.
And then it's also going to be based off of the Lab 11 lecture. So Lab 11. So first thing would have been checking out the results from last week. So it's all the growth of our different organisms on our McConkey agar. So for Lab 11, we're going to be checking out how microorganisms can survive in aquatic environments and how we test for these microorganisms. So for the objective that we have qualitative and quantitative testing methods. We'll go over those. We have our microbiological limits, importance of coliform detection, membrane filtration, serial dilution, standard plate count. Okay, so first off, what is a coliform? The coliform is a gram-negative, rod-shaped bacteria. They're going to be typically enteric or gut bacteria, so they're based in the digestive tract. They're going to be flagellated, so they're going to have flagella, lactose fermenters, and they will not form spores, okay? No spores. So these are going to be really good indicators of sanitation in both food and water. So if there's a lot of coliforms in the food, then that means that the food or water does not come from very sanitary conditions. So there's two different types of testing for coliforms. There's qualitative and quantitative. So qualitative testing is just testing for positive or negative presence, so the presence or absence. And then quantitative is actually going to be counting, so quantity of coliforms present. So for our drinking water, the Safe Drinking Water Act is going to be enforced by the EPA, and this is what is the standard for drinking water. So drinking water needs to meet this standard in order for it to be considered safe. So for drinking water, we have a zero tolerance for coliforms. So that means that there's 0 CFU per ml in drinking water. And then for non-drinking water, there needs to be no more than 100 CFU per ml. Okay. And the reason why we want to test for these is because these are going to be coliforms are going to be an indicator of possible intestinal pathogens that can be contaminating our water. So the water treatment process, we have our wastewater, which gets screened, then goes through a sedimentation process. So whether it's soluble or insoluble, if it's insoluble, then it'll become a sludge, go through this anoxic digestion, and then if it's soluble, it'll go through oxidation. Okay, so you don't have to know any of this too much. I'm not going to be quizzing you on this part exactly. So for this, our standard plate count, also known as an SPC, is going to allow you to determine the number of viable. So viable means the cells that are bacteria that are can survive and reproduce. So they're also going to be testing for heterotrophic, non-aerobic bacteria in a sample. So it's going to be useful for monitoring the efficiency of your different various water treatment processes. So just how well it can get rid of the viable organisms. Okay, so for these procedures, materials, you don't need to know these too much. But I just wanted to show you real fast how these dilutions work. These are called serial dilutions. And so how it works is you take 1 milliliter of your original culture, you place that into a 99 or 9 milliliter broth depending. So it's going to give you a dilution, and then you take 1 ml from there and you add that to another tube. You take 1 ml into there and it just keeps diluting it. So this is going to be 1 ml into 9 milliliters to give you 10 to the negative 1 dilution. Okay. So For the MPN method, or most probable number, this is going to be a statistical based method. So it's not going to give you an exact count, it's going to be more of an estimation. And so you're going to have to use the MPN table in order to determine the amount. So you're going to be observing for turbidity, gas, and the color of the tube in order to determine whether it's positive or negative. So in this case, the yellow color is a positive result and the pink color is a negative result. So we have our tubes that have 10 milliliters of our original sample, our tubes that have 1 milliliter of our original sample, and then our tubes that have 0.1 milliliter. So the more liquid, the more milliliters from their original sample they have, the higher the chance they have of being positive, the chance of having the organism inside of the sample. So with this particular example, we have 5 positive tubes at the 10 ml, 2 positive tubes at the 1 ml, and then 0 positive tubes at the 0.1 ml. So, so you just use this table real fast. So you use this table in order to figure out how many, what's the most probable number of coliforms in your sample per ml, per 100 ml, okay, per 100 ml. So for our example here, we have 520, and if we're to look, then we have 5 at the 10 ml, so that's the 10 ml is this column, or this column. So we'd look through, it goes up to 4, so now we need to go over to here, and this is where the 5 positive results. So then we know that there's 2 positive and 0 in the 1 ml, right? So 520, that is going to give us 50 MPN, 50 coliforms per 100 milliliters. Okay, so that's how you have to do it. So for our membrane filtration method, this is kind of shown in the video that I posted for all of you to check out. It's also going to be on the quiz. So if you have E. coli, they're going to be shown up as blue-green colonies on here. And then if you have other fecal coliforms, they'll just show up as a purple colony. Okay. So with that, we have some more procedure information, but I don't need to worry too much about this. So that's it. That's all you're going to need to know for your quiz. Just make sure to figure out all that stuff and take the quiz. And that's it.